Hey, Hammy here, back with part 2 of section 3.1 on our introduction to cells, where we want to take a look at different kinds of microscopes, limits to cell size, and also about cell shape a little bit. So let's get started. First, looking at light microscopes. Okay, light microscopes are, we call them light microscopes because they use light. So you see down here is the light source. It shines up through the specimen, up to our eyes. So you have to have something that you are able to see through. For example, a lot of thin slices of tissues or cells, those kind of things. I like when people put their finger up under the microscope and they try to look at it. Well, light can't pass through your finger, so it's just going to look dark or like a pencil or something. It has to be a thin specimen. For the light microscopes we use in lab, and they have a magnification up to about 1,000x, 1,000 times bigger. Sometimes if you want to look at something that light doesn't pass through, like this yellow jacket over here, you place the yellow jacket on a stereo microscope. The light source is here. It bounces off the specimen and then up through your eyes. So we can look at things. We get these nice, cool pictures like this 3D specimen down here. Yeah, how do I clear it? There we go. Sorry, learning a new program here. The other kind of big microscope we look at is this idea of electron microscope, which uses beams of electrons. Their magnification is a lot bigger, up to 1,000,000x on the really expensive ones. Other ones are a couple 100,000x. And they often will take up a whole room. When I was in college, we had one. I never used it, but I remember seeing it as kind of in a small room in the science department. And they're usually connected to a computer. Unlike a light microscope where you can look at living cells, living things, living tissues, electron microscopes you cannot, because the transmission electron microscope over here shoots beams of electrons through the specimen, and you can see really tiny things like organelles of a cell. The scanning electron microscope scans over the surface. Oftentimes you have to plate it in gold or something shiny, but we get these cool little three-dimensional type pictures like this. So they're really expensive. They're tens or hundreds of thousands of dollars. Not everyone can afford those kind of microscopes. When we look at the diversity of cells, your book mentions just, again, a lot based on the characteristics of life. Because if life is made up of cells, they're going to use, they're responsible for getting and using energy, responding to the environment and changing conditions around them. And also, cells will reproduce and make more cells. Okay, their functions and shapes we'll get into a little bit more in here in a second. But if you look at, these are all muscle cells, but look very different because they have slightly different functions in our body. Cell size. Why do we need microscopes to see cells? Why are they so small? Okay, there is this idea that the cell needs certain nutrients to survive. It needs to get rid of waste. How does it do that? Well, we look at the surface area. Okay. Ooh, this can be really rough with this program, so hang with me. And this is really hard with the trackpad. Surface. Well, you know how to spell area. Surface area to volume. The surface area of the cell represents the area around the cell. Okay, so here you have what would be the cell membrane. Okay, so here you have a 1 by 1 by 1 cell. So if you take the surface area of one side, 1 times 1 is 1 square centimeter times 6 sides. You get a surface area of 6 square centimeters. The volume, LWH, is 1 times 1 times 1, so you get 1 cubic centimeter. So your surface area to volume ratio is a 6 to 1. So for every unit of volume, you have 6 units of surface area. If the cell grows, so over here we have a cell that's 3 by 3 by, where's the other side? 3. Okay, so one side is 3 times 3 times 9 times 6 sides gives you a surface area of 54. The volume, LWH, 3 times 3 times 3, is 27. So you have 54 to they both got bigger. It should have more surface area. It does. But surface area per volume, if you look at the ratio, 54 divided by 27 is 2, so 2 to 1. So a small cell had 6 units of cell membrane for every unit of volume to get things in and out, whereas a large cube, 3 centimeters squared, has only 2 units of cell membrane for every unit of volume. Not very efficient, not as efficient. So as our cells start to get bigger, they will actually divide and just make more smaller cells than being made up of a lot of big cells. And finally, when we talk about cell shape, you'll notice, remember I talk about F cubed all the time, form fits function. Okay, so you see all these different shapes. That helps them do whatever their job is. Red blood cells here, okay, have kind of this donut shape, which makes them very flexible. So when they go through your little tiny capillaries, they're able to bend and flex. Bacteria helps them stick to your intestines in different places. Pollen grains helps them stick to things, so they're spread on the little bee leg from one flower to the next. Algae have these little flagella that help them swim through the water. If you look at a nerve cell, over here's kind of a bigger picture of a nerve cell. What do? They're like the wiring of our body, right? Transmit impulses from organ to organ. So you see they have these long extensions called axons, and they're highly branched at each end so they can connect to each other. Okay. Over here on the plant cell, plants need to breathe, right? They need CO2, carbon dioxide, for photosynthesis, and they get rid of oxygen, but they don't have lungs like us. But they have these little holes, these 2 cells on the surface of their leaf that create holes or stomata so that the air can enter and leave the leaf for photosynthesis. So cells have very specific shapes based on what function they provide. Okay, all that factors into what we call homeostasis and helping the organism, the plant, the animal, the fungi, the paramecium, the protist, all functioning, okay, at kind of these stable levels and helping them to survive. In the next part of the chapter, we'll start to get into specific parts of the cell, and we'll look at some of these things that help the actual cell to function.